understanding the genomics of speciation because we have a huge amount of new data that's allowing us to describe sort of the patterns of speciation. So what extent are genetic differences between species, and how parallel are these differences across lineages? But we still have a lot of questions remaining about the processes of speciation, including what is the role of natural selection in driving these patterns, and how are these patterns affected by intrinsic properties of the genome, affecting the process of differentiation and speciation. So if we consider a reproductive isolation loci, we can use some common metric like FST, which is a measure of genetic differentiation, to look at the differences between these loci in species A and species B. But what's been increasingly appreciated is that this may not be quite sufficient because FST is based upon variance in allele frequency between species and the variance in frequency of alleles within populations. So as a result, sort of other selective processes that change variance in allele frequency can also lead to elevated genetic differentiation, such as sort of background selection or link selection. It's also been increasingly appreciated that components of genomic architecture, such as recombination rate variation or gene density, can also affect FST. So combine this gives the effect that elevated genetic differentiation is consistent with the reproductive isolation loci, but by itself is not enough evidence to conclude that the loci is involved in reproductive isolation. So we can also consider another genetic metric, DXY, but this is a measure instead of genetic divergence, which looks at the average differences between site between two species. And in this case, we can see, so that much older loci would have a much larger DXY, where newer loci that changed in response to something like linked selection would have a lower DXY. So we can kind of combine this all together and leading to some expectations of what we would see. Under a scenario of reproductive isolation with gene flow, we would expect to see lower nucleotide diversity and elevated FST, but this would be combined with high DXY. And alternative scenarios of sort of positive selection or background selection, we would see the same patterns with nucleotide diversity and FST, but instead a decrease in DXY. And what I would also want to point out is that in this category, we could still find loci that lead to reproductive isolation, but that involved instead in response to selection and allopatry. So I'm going to consider this group of paper wasps. So they are a primitively used social species. They make these open paper nests, and they're super common kind of across North America, but I'm going to talk about 3 species. So Callistes fiscatus, which has this distribution in blue, Callistes metricus, and Callistes dorsalis. So as you can see, these species are sympatric across large portions of their ranges. They also nest, forage, and mate in similar habitats. So they're really sympatric. Fiscatus and metricus are quite a bit more closely related to each other, where dorsalis is an outgroup, and they're also really genetically distinct. It also previously shown in a mate choice experiment that the more closely related Fiscatus and metricus are mostly but not entirely behaviorally isolated from each other. So I think that this makes a really interesting group in which to consider sort of the role of the genomics of speciation. So to do this, we did whole genome resequencing of individuals collected from the 3 species from at least 2 populations. So 40 Fiscatus, 19 metricus, and 18 dorsalis. So all of this data was aligned to the Fiscatus genome for the slides that I'm going to show next, but I want to point out that all of my conclusions also hold true if you align them to the alternative genomes. Okay, so first I'm going to show an FST plot for Fiscatus and metricus, and I'm just going to show the first 4 chromosomes just for simplicity's sake. So here we can see that there is this pattern of heterogenic genomic divergence, with some areas of the genome showing really high areas of FST, and other areas of the genome sort of showing lower areas of FST. So if we compare this for other FST plots, for Fiscatus and dorsalis and metricus and dorsalis, we see sort of an overlap of these outlying peaks and also an overlap in these outlying valleys. But also, for our more diverged species comparisons, this Fiscatus dorsalis and Metricus dorsalis, the overall level of FST is higher. And what I think is also interesting is that we also plot on this an FST comparison instead of between species, between populations. We actually also see some overlap between these outlying windows. Okay, so to do a more formal analysis, so now I'm showing data for the entire genome. Each dot is going to be a 50 kilobase window. And I'm going to show all combinations of pairwise comparison of FST. So for example, the far comparison, the x-axis shows Fiscatus dorsalis FST, or the y-axis shows Fiscatus metricus FST. So overall, these are pretty highly correlated, so high FST regions are shared across species. But if you're a stickler, you could point out that the same genome is used on both axes in all of these comparisons. So I'm going to throw in a bonus fourth species and show that this also holds true when none of the genomes are the same. So I wanted to see if we could say sort of anything about why we're seeing this shared pattern of high FST. And to do this, I wanted to look at the relationship between DXY and FST in these groups. So now I'm showing FST on the y-axis and DXY on the x-axis. And just a reminder that our expectation for loci that would be involved in reproductive isolation gene flow would be that they'd have both high FST and high DXY values. But we see that there is a negative correlation between these for all of these comparisons, and particularly for our more closely related species, Fiscatus and Metricus, there's really nothing in this high FST, high DXY area. So together this is sort of a pattern that would be consistent with shared linked selection, but we can't really say much about whether or not this shared selection is sort of positive selection or the result of background selection from what we have here. So I wanted to see if I could tease out of the contribution of background selection, and to do this, I took advantage of a nice feature about social hymenopteron, and this is the fact that they're haplodiploid. So if we consider a lethal, deleterious recessive mutation in a female, these mutations will be masked from selection and she'll be just fine. But these same mutations in a male will be immediately removed from the population. So based on this, we can, So based on this, we can make a prediction that if we're going to look at a population of females, that their overall genetic diversity should be higher than male genetic diversity as a result of this removal of deleterious alleles in males. So I wanted to actually test this. So using this female Fiscatus data, we combined it with additional whole genome sequencing from 24 males, and then calculated a measure of genetic diversity. In this case, this is Waterson's data for both males and females. And due to the difference in sample size, I just scaled this so we can compare between them. And our expectation based on the predictions is that females should have greater nucleotide diversity than males, and this will be evidenced by a positive value of this trait. And this is actually exactly what we see. So I'm just going to show this in two ways. So this is a histogram of this delta theta value, and the dotted line here is our mutual expectation of 0. And then also I'm showing this on a plot over here against FST. And so we can see that almost entirely that this value is positive, matching our expectation that females have greater nucleotide diversity than males. But what's even more interesting is that there is a significant positive correlation between this value and FST, suggesting that males are more under selection in regions with high FST, which I think is also suggestive that maybe background selection is higher in these regions as well. So briefly, I wanted to say something maybe more positive. So I am adding an additional metric, this AN-RUS-H, which is just a modification of Tajima's-D that uses an ancestral genome to put things in context. I'm not going to go too deep into that, but we can, kind of using the same idea, come up with an expectations of what we'd expect for positive selection. We can find sort of this space for only those regions that have high FST values. And this seems to be a pretty promising area because within these windows are some exciting genes that have been linked to reproductive isolation or sort of testes and gonad development in other organisms. So in general, speciation in Plistes is, I think this is a really cool system that I would love to talk with anybody about later. They're an incredibly diverse, colorful group. But instead I'm going to actually tell you about a cryptic divergence project that I'm also working on in Plistes metricus. So this is just a sort of representative sample of pin specimens from a museum in the species. So what we did is we collected and sequenced 226 metricus females from throughout the eastern United States. And when we put them into a principal component analysis, to my surprise, we popped out two distinct clades. And just in case you're worried about this affecting the results that I talked about before, everything I talked about before was actually just the metricus 1 clade. If you map this back onto their distribution, we see maybe what's suggestive of sort of an Appalachian area divide-ish. But there are also regions in which these two distinct groups occur together in symmetry. So kind of doing similar things, if we do a FST scan between metricus 1 and 2, we find same high and low divergence regions. But we actually see a very different DXY plot with almost no correlation between FST and DXY, and some regions that definitely have shared FST outliers and shared DXY outliers, suggesting that maybe different processes are driving divergence in this group than divergence in the older clades. So just to kind of sum up, Plistes that showed shared patterns of heterogenic genomic divergence. These are consistent with linked selection, but using the magic of haploidiploidy, I think I can show that at least some of these are the result of background selection, and that this background selection is stronger in regions with high FST. There's an exciting new lineage that I'm interested to explore, but know very little about, so stay tuned in the future. And different mechanisms may drive ancient research diversification. I just want to thank Mike Sheehan and other members of the Sheehan Lab and people that helped with wasp collecting and the funding sources. And I think I have time for a really quick question.